Welcome to the video tutorial series on genomic tumor testing patient reports, brought to you by the Genomic Education team at the Jackson Laboratory. This short video will provide an overview of the patient report and walk through some of the patient demographic information. In this video tutorial series, we will walk through sections of a sample patient report detailing the outcome of next-generation sequencing on tumor tissue. Somatic tumor testing provides clinicians with information to help them formulate a treatment plan tailored to a specific patient based on their genetics. The information included on this type of report is a list of clinically significant gene variants identified through sequencing and a list of FDA-approved drugs or clinical trials that are associated with each gene variant and cancer type. At the end of the report, there is fine print detailing the methods taken to acquire the data on the report, as well as a disclaimer discussing the limitations of sequencing and the potential caveats to the data presented in the report. Here we have a report for Tony. Tony has melanoma, which is a type of skin cancer. At the top of the report, we can see some demographic information about Tony and the sample that was subject to sequencing. All patients are given a patient ID number to conceal their identity, but this report does include some identifying information, such as date of birth, as well as assigned biological sex, which are listed in this first column. Next, in the middle column, we have information about the specimen collected for sequencing, including the specimen number and specimen source ID. The next field is the primary site of the tumor or the origin of the cancer in the body, which in this case is the skin. For Tony, the specimen was also taken from the skin, which is shown under specimen site. These locations do not always have to match. If the tumor has metastasized to another location, the specimen could be taken from the site of metastasis, which would then differ from the primary location of the tumor. The type of biopsy sample here is FFPE, which stands for formalin-fixed paraffin-embedded tissue, which is a way to preserve a tissue through fixation and embedding, which locks everything into place before analysis. Without fixation, tissues would begin to degrade once taken out of the body. The neoplastic content is an estimate of the percentage of the sample that is comprised of tumor cells. And finally, there is a date the sample was received at the location for sequencing. In the right column, there is contact information for the physician who collected the sample. Thank you for listening. For more information on genomic tumor testing patient reports, continue watching the other videos in this series. Also, check out the other resources provided by Jack's Genomic Education on our website.